Hi students, in the last class we discussed about some of the fruits. DNA is the genetic material, right? And we discussed about two experiments. One is the transmission principle and next one is the bacteriophage experiment, Hershey and Chase experiment, right? And coming to this class, we are discussing about how the DNA replication takes place. How the DNA replication takes place. In the last class, along with that experiments, we discussed about RNA and why RNA not existed like genetic material. Means in initial cells, RNA is the genetic material. Later, RNA contained cells are replaced by DNA contained cells. Why RNA not existed in the form of genetic material? Why RNA replaced by the DNA? Why RNA replaced by the DNA? We discussed, right? And still RNA showing the dominancy by the splicing mechanism. We already discussed, right? So that is about that last class summary. And coming to this class, we are discussing about the DNA replication process. In that case, autocatalytic function. DNA majorly showing that autocatalytic function. Here autocatalytic function is nothing but synthesis of DNA from DNA. Synthesis of DNA from DNA. That is called as autocatalytic function. Right, so this is also called self-replication process. DNA have the capacity to replicate by itself because a group of enzymes are involved in the DNA replication process. So self-replication exhibited by the DNA. Right, so here autocatalytic function of DNA. Here synthesis of DNA to DNA. That is called as DNA replication process. Right, and coming to the next one. Or suppose we are discussing about here eukaryotic cell. Just imagine this is the eukaryotic cell. Means either in prokaryotic cells, either in eukaryotic cells, the DNA replication takes place. But here we are discussing about interphase, right? So interphase that is seen in eukaryotic cells. In the last year we discussed about the cell cycle. Within cell cycle, two phases are there. There is an interphase is there, right? And other one is that there is an mitotic phase is there. See, we discussed about karyokinesis process, right? Cytokinesis process. We discussed in the last year cell cycle. That is a chapter, right? So here, what I am telling here, during interphase there is an S phase is there. S phase indicates here synthesis phase. Within S phase, the DNA synthesis takes place. Within S phase, DNA synthesis takes place. So within S phase, the DNA synthesis takes place. This is the one of the important phase for the cell cycle because in this phase DNA replication takes place. After the S phase, there is in G2 phase is there. First phase is G1 and next one is the S phase and next phase is the G2 phase, right? After that G2 phase here followed by means karyokinesis process. Karyokinesis is nothing but division of genetic material. Division of genetic material. That is nothing but separation of chromosomes takes place. Separation of chromosomes takes place. After that chromosome separation process, there is an cytokinesis takes place. Cytokinesis takes place. So what I am telling here in the normal cell cycle process, there is in G1 phase, S phase. S phase is for the replication of DNA, right? And G2 phase is there. And next one is that there is in karyokinesis process takes place. And there is in cytokinesis process takes place. Okay, once the cytokinesis takes place in the regular condition, one cell divided into two cells. One cell is divided into two cells. One cell is divided into the two cells. In the case of the mitosis process, we are not discussing about here meiosis process. Again, students got the doubt here. That's why I am saying here. This is the just imagine this is the mitosis process. So here one cell is divided into two cells, right? In certain cases, if karyokinesis not takes place, for suppose this is the one of the cell, and this is the nucleus, and this is the initial genetic material. Okay, this is the genetic material, this is the DNA, right? After S phase, one copy is doubled. Generally, cell, this one is the one of the body cell 2n state, and this is also another 2n state. Okay, see if the division takes place, one cell get that 2n state, one set of chromosomes. Here one set means 2n state genetic material enter into the one cell, another 2n state genetic material enter into the another cell. Then after that the one cell divided into two cells. So there is no problem if the cell division takes place, no problem. But in certain cases okay, karyokinesis is absent. If karyokinesis is absent, what happened? See, the condition is called as polyploidy. The condition is called as polyploidy. Generally, okay, we have seen there is an haploid state. Haploid state is there, and in certain cases, diploid state is there. Okay, generally mitosis condition, diploid state only. Haploid, that is seen in meiosis condition, right? So here, if S phase is completed, if cells are not undergoes the karyokinesis process, then inside the nucleus, okay, more than 2 sets of chromosomes are present. More than 2 sets of chromosomes are present here. So the condition is called as polyploidy. The condition is called as polyploidy. Okay, here again these genetic material involved in the S phase. If again the separation is not takes place, again genetic material is increased. The genetic material content is increased. So the condition is called as polyploidy. If inside the nucleus genetic material content is increased, the condition is called as polyploidy. Okay, generally karyokinesis takes place, no problem. If karyokinesis is absent, then the cell contains the nucleus. The condition is called as polyploidy, right? And cytokinesis. If cytokinesis, okay, in certain cases, this is one of the cell, right? This is one of the cell. Here there is a karyokinesis takes place. One nucleus is divided into the 2 nucleus. This is the 2 end-state genetic material. This is 2 end-state genetic material. But here cytokinesis is absent. If cytokinesis is absent, the condition is called as syncytium. The condition is called as syncytium. Syncytium is indicates here. See, multinucleated condition. Multinucleated condition. One cell is there, but inside the cell, maybe the 2, maybe the 3, maybe the 4, maybe the 6. So many number of nucleus are present. The best example is tapetum. We discussed in the first year, right? Means whatever that plant anther walls are there, and within anther walls there is a tapetum tissue is there. The tapetum tissue having that multinucleated condition, binucleated condition or multinucleated condition. So if the cytokinesis not takes place, the condition is called as syncytium. If karyokinesis not takes place, the condition is called as polyploidy. So that is the difference between the karyokinesis and the cytokinesis. Karyokinesis, yeah. If karyokinesis not takes place, polyploidy. If cytokinesis not takes place, that is syncytium, right? So that is about general discussion about that cell division process. Yeah, coming to the next one. Here we are discussing about the DNA replication process. There is models of different models of DNA replications are there, because in the olden days the scientists are not having that proper equipment. That is the reason why the scientists proposes some theories how the DNA replication is take place. We know that DNA is the double helical structure. Right? So based on that, that time maybe scientists are knows about that DNA replication or DNA structure. We don't know, but some theories are proposed. So this one is considered as that models of DNA replication process. So the very first one is called as conservative model. Conservative model means, for suppose this is the one of the DNA. Right? So here this is the double helical structure, no doubt about it. Here this is the double helical structure of the DNA. After the DNA replication process, this is the parental DNA. As it is, this is the parental DNA. This is the parental DNA. And newly synthesized DNA. So this one is newly synthesized DNA. So what I am telling here, whatever that parental DNA is present, after DNA replication also, the parental DNA enter into the one cell. The complete parental DNA enter the one cell, and newly synthesized DNA enter into the another daughter cell. Remember all of you what I am telling here. According to the conservative model, this is the parental DNA. After DNA replication process, right? That parental DNA enter into the complete parental DNA enter into the one daughter cell, right? And newly synthesized DNA enter into the another daughter cell. Generally, this is the model. This model is called as conservative model. Completely parental DNA, conserved means separated and enter into that one daughter cell, and newly synthesized DNA enter into the another daughter cell. So this is the failure model. This is the failure model. Conservative model is the failure model. Why conservative model is the failure model? Because see, during the DNA replication process, this is the parental DNA. This is the parental DNA. Okay. During DNA replication process, the parental DNA is separated. This is the parental DNA and this is the parent DNA. This is the separation process. After the separation process, right, and this is newly synthesized strand. This is newly synthesized strand, and this is another newly synthesized strand. This is another newly synthesized strand. So, generally, after DNA replication process, one strand is belongs to the parents and another strand belongs to the newly synthesized strand. One strand belongs to the parental DNA and another strand belongs to the newly synthesized DNA. So this one is possible. This one is possible. Say completely after DNA replication process, the DNA entered to the one parental cell, the complete DNA entered to the one parental cell, one daughter cell, and newly synthesized DNA entered to the another daughter cell. This is not possible. So this one is possible because once the two strands of the DNA is separated, the double helix structure, once the two strands are separated and another strand is newly synthesized. Here also another strand newly synthesized. This one is possible. This one is possible, right? So that is about process of the DNA replication process. And coming to the next one is the semi-conservative model. According to this model, right, this is the parental DNA. This is the parental DNA, and after DNA replication process, right, this is the parental DNA, this is also parental DNA, and this one is newly synthesized strand. This one is the newly synthesized strand. This one also newly synthesized strand. So what I am telling here, this is the exact model. This is the exact model, and most of the scientists also accepted the semi-conservative model is the best model for the DNA replication explanation. And this is also one of the best evidence, but there is one more part is added to that semi-conservative model. I will explain later, but up to this part, according to this model, the parental DNA separated for the DNA replication process. So in daughter cell, one strand belongs to the parental DNA and another strand is newly synthesized. Here also same thing, one strand belongs to the parental DNA and another strand newly synthesized. So this is the way for the DNA replication process. So that is about the semi-conservative model. According to that dispersive model, see, this is the parental DNA. Right? And the parental DNA in the next generation, this is the bit and this is another bit. And like this means here this is the bit belongs to the parent DNA and this is the bit belongs to the newly synthesized DNA and this is another bit, this is parent DNA and this is means here reverse. So here like this means exactly this is the newly synthesized DNA and this is parental DNA and again this is newly synthesized DNA, right? This is parental DNA. So what I am telling here, exactly by this model, a bit of DNA, this is a bit of the DNA. This is a bit of the DNA. This DNA belongs to the parent DNA and another bit is newly synthesized. Again another bit belongs to the parent DNA. Another bit is newly synthesized. So this is the bit belongs to the newly synthesized DNA and another bit is the parent DNA. So here blue color indicates here parent DNA and the red color indicates here newly synthesized DNA. So what I am telling here, the DNA synthesized in bits manner. A bit part or a part of the DNA belongs to the parent DNA and a part belongs to the newly synthesized DNA. Dispersive. In the dispersive manner DNA is synthesized. So this is one of the failure model. Because in this manner DNA is not synthesized. Because always one strand belongs to the parent strand and one strand is newly synthesized. But what I am telling here, okay, complete DNA is there. In the DNA, a bit belongs to the parent DNA, a bit is newly synthesized DNA. That is not possible. That is not possible. That's the reason why here dispersive model is also failure model, right? So that is about different models of DNA replication process. We'll discuss about the evidences for the semi-conservative model. Again, in this one also one more topic is there. I'll discuss now. Here coming to the next topic is that semi-conservative model, and this one is the experimental evidence for the semi-conservative model. So just we discussed conservative model, semi-conservative model, and there is a dispersive model. See, this is the experimental evidence. So before going to the experimental evidence, the history and who are involved, means the names of the scientists which are involved in the semi-conservative model experiments. So this was first proposed by the Watson Crick. See, semi-conservative model was first proposed by Watson and Crick, right? And it was proved by the Michelson and Stahl. Michelson and Stahl experiment. They did the research on E. coli. So remember, Michelson and Stahl, they do not used, means they didn't use radio-labeled substances, right? They used heavy isotopes, N15, right? They used the heavy isotopes, but they did not use radio-labeled substances, right? And chromosomal, yeah. In eukaryotic cells, semi-conservative model, okay, they did, the scientist name is the Taylor, okay, did the research on Vicia faba. The plant name is the Vicia faba, right? That is the plant name. This is one type of bean. We know that one of the Fabaceae family, right? Vicia faba. That is one of the bean plant, one type of bean plant. And Taylor is a scientist. He explains the semi-conservative model, especially in chromosomes, the chromosome replication done by the semi-conservative model, proven by the Taylor, but he used radioactive thymidine. So what I am telling here, eukaryotic cells, semi-conservative model, okay, experimental evidence, okay, the scientist used the thymidine, radio-labeled substance. That is the radio-labeled thymidine substance. See, what I write here. Radio-labeled thymidine. So thymidine is used by the scientist. Thymidine, that is especially radio-labeled thymidine, that is used by the scientist. But Michelson and Stahl, they used heavy isotopes. So that is about that the sources of the different experiments, and exactly DNA replication is the semi-conservative discontinuous model. Exactly, DNA replication is the semi-conservative, discontinuous model. Means what? I said semi-conservative model, and one more term is added to that DNA replication process, that is discontinuous. Semi-conservative discontinuous model means what? See, this is the one of the parental DNA, 5' to 3' and 3' to 5'. These are the parent strand. The black color strands are indicates parental strands, and the blue color, this is the blue color. The blue color one is indicates here newly synthesizing strand. So during DNA replication process, one of the strand is continuously synthesized. See, this strand is called as leading strand. And one of the strand is discontinuously synthesized. So see, one strand is continuously synthesized, another strand is discontinuously synthesized. So DNA replication is the semi-conservative discontinuous. So discontinuously, one strand of newly synthesizing DNA discontinuously synthesized. So these fragments also called Okazaki fragments. These are also called Okazaki fragments. So in DNA replication process, the exact model is semi-conservative discontinuous model, because one of the strand is continuously synthesized but another strand is discontinuously synthesized. That's the reason why this is called as semi-conservative discontinuous model. Semi-conservative discontinuous model, right? So that is about the DNA replication process. And we will discuss the experimental evidence, Michelson and Stahl experiment now. Coming to the next topic is the Michelson and Stahl experiment, and according to this experiment, see, this is the one of the evidence for semi-conservative model. Especially this process means the Michelson and Stahl experiment done in E. coli cells, right? This research done on prokaryotic cells, right? In the last topic we discussed about Visya Fava, right? The chromosomal replication explained, okay. One of the Taylor, scientist name is the Taylor. So that is the eukaryotic cells replication process, right? That is also confirms that semi-conservative model. This one also confirms that semi-conservative model. This model done in E. coli cells, right? And see, for suppose here the very first parental bacterial cells are there. Very first, this is one of the parental bacterial cell culture. Okay, up to several generations. Up to several generations or up to many generations, the bacterial cells which are grown in N15 media. The bacterial cells which are grown in N15 media. Remember here, two types of isotopes are used. One is N15. Other one is N14. First of all, remember these are not radiolabeled substances. N15 and N14, these are not radiolabeled substances. These are heavy isotope. See, N15 is the heavy isotope and N14 is the normal isotope, right? No, sorry. Yeah, N14 is the normal one. N15 is the heavy isotope, right? So here what I am telling, here nitrogen, that is nothing but N15 contains media. Within that media, up to several generations, the bacterial cells are cultured. So once the bacterial cells are cultured up to several generations, so both strands of the DNA, okay, the both strands of the DNA becomes N15 and N15. N15 and N15. Sir, where that N15 utilized? How the N15 present within the DNA? We know nitrogen bases are there. For the nitrogen basis synthesis, nitrogen is utilized. So for that nitrogen basis synthesis, the N15 is utilized. Automatically the nitrogen getting the heavy weight. Sorry, the nitrogen having the heavy weight. Nitrogen bases are getting the heavy weight, right? Because presence of N15, DNA having high molecular weight. Presence of N15, DNA having high molecular weight. So that is about N15 and N50. So after several generations, means after many number of generations, right? The both strands of one of the bacteria contains N15 and N15. So this is the parental generation. This is the one of the parental generation. Very first generation is the parental generation. So both strands are here, N15 and N15. Once N15 and N15 strands are synthesized within that bacteria, right? The bacteria cells are collected and that bacteria cells are subjected for the degradation process. Once the degradation is completed, right, and whatever that whatever the contents are present, these are subjected for the density gradient centrifugation process. Whatever the contents which are collected from the bacteria cells, which are subjected for the density gradient centrifugation process. Especially this is the test tube. This is the test tube, right? The test tube. This is the top part of the test tube. Right? So inside the test tube, many number of layers-like structures are formed. By the usage of cesium chloride. So what I am telling here, cesium chloride is utilized for the preparation of density gradients. Inside the test tube, okay, there is a density gradient is prepared. That density gradient prepared by the usage of cesium chloride. What is the use of that density gradient preparation? See, inside the test tube, if we add any molecules, which are separated based on molecular weight, For suppose, if we add one molecule here, okay, See, if we add two types of molecules here, some are present at here, and some are present here, and some are present here. Okay, if we add a group of molecules which are separated based on molecular weight. Okay, so this one is indicates high molecular weight is present. This one is indicates medium molecular weight is present. This one is indicates low molecular weight is present. So what I am telling here, molecules are separated based on the molecular weight. Molecules are separated based on the molecular weight. So that is the use of cesium chloride. That is the use of cesium chloride. So we have collected one of that N15 and N15 DNA, right? That DNA band formed at the bottom side of the test tube. So this one is indicates N15 and N15. This one is indicates here N15 and N15 band. This band is N15 and N15 band. N15 and N15 band. That band formed at the bottom side of the test tube. Bottom side of the test tube. Right? So how can we identify that DNA band? Generally we can't see that, we can't see the cells, we can't see the DNA. Then how can we identify the DNA band within the test tube? Within the test tube. See, we are using some of the dyes are there. The dyes are fluorochrome and ethidium bromide. Fluorochrome and the ethidium bromide. Both are the dyes which are using for the identification of band or visualization of the band. So if we add that fluorochrome and ethidium bromide substances, these are the dyes. If we add to the test tube, then the bands are emits the light. Especially here, the bands are produces the color. The bands are produces the color. Generally, the color is the orange color. The color is the orange color. So here, this is the band. The band, okay. The band produces the one of the color presence of dyes. The band produces the colors presence of dyes, fluorochrome and the ethidium bromide. So once the color is appear, we can confirm the band is the DNA band, because these molecules are specifically bind to the DNA. The molecules are specifically bind to the DNA. So that is about the first generation or parental generation. Not first generation, that is the parental generation. That is the parental generation. Coming to that first generation, see, this is the first generation cells. See, whatever that parental cells are there, the parental cells are taken out and which are introduced into the another type of culture media, because here very first generation the nutrient media contains N15, and coming to that next generation, this is the next generation nutrient media, it contains the N14. The nutrient media, the next generation nutrient media contains the N14, right? So what I am telling here, this is the parental generation and this is the first generation. And if we culture the parental generation cells in N14 media, if the parental generation cells are cultured in N14 media, then how the DNA synthesis takes place here? See, according to that semi-conservative model, one strand enter into the one cell. Okay, this strand enter into the one daughter cell, and this strand enter into the another daughter cell, and another strand. Okay, another strand of the daughter cell. Okay, this one is newly synthesized. Here also another strand of the newly synthesized DNA. Okay, in daughter cell. This is also newly synthesized. So this one is newly synthesized. This one is newly synthesized. Okay, here the source is N14. Here the source is N14. The new cells are cultivated in N14 media. The new cells are cultivated in N14 media. So what I am telling here, whatever that parental strands are there, the parental strands are N15 and newly synthesized strands are N14. The newly synthesized strands are N14, right? So that is about that first generation cells. That is about the first generation cells. Exactly, the cells are cultivated here, another media for 20 minutes, because the bacteria cell, the cell division completed within 20 minutes, right? So after that 20 minutes, the cells are collected and the cells are digested or degraded. The cell walls are degraded, right? That fluid is taken out or centrifuged, and that is subjected to the density gradient centrifugation process. That is subjected to density gradient centrifugation process. So this is one of the test tube. This is one of the test tube, right? So here cesium chloride is present. As usual, the story is cesium chloride is present. That is the density gradient centrifugation process. And if we separate the DNA molecules, this generation, right? The first generation DNA molecules are separated within density gradient centrifugation process. The band formed at the middle. The band formed at the middle. Sir, why here band formed at the middle? Because one strand is N15 and the strand is N14. One strand is N15 and the strand is N14. Here why band formed at the lower side or bottom side of the tissue? Both strands are N15. So heavy molecular weight. So this is intermediate molecular weight. Intermediate molecular weight. So the band is here N14 and N15. N14 and the N15. The bond formed at the middle of the tissue because these are hybrids. One is heavy, other one is the light. So that is about that first generation cells. Again, the same cells, one of the parent cell, and again the next generation cultured in N14 media. Again the second generation. This is the second generation. Again the second generation cells also cultivated within the N14 media. Again the second generation cells also cultivated within the N14 media. So what happens here? See, within this case, one parental cell, this is the, okay, here, N15, N15. Only two strands are there N15 because the very first cells are cultivated in N15. After that, remaining all cells are cultivated within N14. So after 40 minutes, this is after 40 minutes. First one is after 20 minutes. One bacteria becomes two bacteria. After 40 becomes the two bacteria becomes four bacteria. So here N15 is one strand, N15 is another strand. Remaining all are newly synthesized. Here newly synthesized strands are 2. Here newly synthesized strands are 4 because this one is newly synthesized strand and this one is newly synthesized strand and this one is newly synthesized strand and this one is newly synthesized strand. And the cells are cultivated in N14 media. So if these molecules are subjected to the density gradient centrifugation process, the bands are formed like this. So this is one of the density gradient centrifugation process. Here also, here two types of bands are produced. See, this one is, okay, these two are one type of molecules and these two are another type of molecules. So here at the middle of the tissue, when one band is produced, at the middle of the tissue, one band is produced. This is the middle of the tissue, right? So here one band is produced. We know about this one. One is that N14 and another one is the N15, right? And there is an N14, N14 and N14, N14. Another two molecules are there. These also produces the band. These also produces the band. The band formed at the upper side of the tissue. Upper side of the tissue. This is the band, that is N14 and N14. So based on this, we can say, okay, all the DNA in prokaryotic cells, the DNA synthesis is carried out by the semi-conservative model because this is the first band formed at the bottom of the tissue. The second band, one is that N15, other one is the N14. The band formed at the middle of the tissue. In the second generation, okay, both the bands are formed. One is that, one of the band formed at the middle of the tissue, and next one there is an N14 light color bands which are formed at the upper side of the tissue, or top side of the tissue. So this is the one of the evidence for the semi-conservative model. Remember, some questions are asking sometimes here, if within the parental generation, the N15, the bands percentage is N15, okay, the strands percentage is the 100% because both the strands are N15 and N15. So 100% of N15 strands are there. And coming to the first generation, so here N15% is 50%. And 15% is the 50% because another newly synthesized strands are there. These are N14 strands are 2. N15 strands are 2. That's the reason why N15 is the 50%, N14 is the 50%, right? And coming to the third generation, here N15% is the 25%, N15% is the 25%, and N14% is the 75%, and 14% is the 75%, right? So that is about that evidences for the semi-conservative model. This theory was proposed by Michelson and Starr. Remember, in this experiment they used the heavy isotopes, not radioactive isotopes. But in eukaryotic cells, chromosomal replication, that is done on Riccia faba. That is one of the bean, one type of bean. But that process done by the usage of radioactive substances. So this is about the conclusion for the Michelson and Stahl experiment.